Okay, welcome everyone to the last of our speaker series for this semester. Our speaker today is Angela Zivkovic, who is an associate professor at the University of California, Davis. We're really happy to have her speaking here today. She wanted to come in person, but this still works virtually. And as you can see, she's sitting on a beach in Hawaii, like in the talk. Dr. Zivkovic's research focuses on precision nutrition approaches to prevent disease, optimize health, and extend a healthy lifespan. The group is particularly interested in dietary interventions, foods, and dietary components that reduce inflammation, beneficially affecting the gut microbiome and improve the functional capacity of HDL particles. The current focus is on prevention of neurodegenerative disease, particularly Alzheimer's. She's currently working on 3 NIH-funded projects. She's PI of a multidisciplinary R01 from NIA, studying the relationships between glycosylation and Alzheimer's. She's PI on a UC Davis subaward with Johns Hopkins on the NIH Common Fund Extracellular RNA Communications Consortium, and PI on a technology development grant funded by NIGMS to develop advanced technologies to isolate and characterize HDL and other nanoparticles. Dr. Zivkovic also runs a precision nutrition consultation practice and was the technical co-founder of Mimeo, which spun out of her lab, and is a scientific advisor at Realm Foods. Her title is Diet and Nutrition in Precision Medicine, What are the Targets? As again, this is a webinar. If you would like to have questions at the end, put them in the chat and I will ask them right at the end. And thank you, Angela, for giving the talk today.
Well, thank you so much for that introduction and for inviting me to give this talk today. I'm delighted to be here with you all, and hopefully one day I'll be able to visit you in person as well. So I wanted to start by kind of showing you this, this figure that I put together years ago as I was kind of struggling with trying to understand why people's responses are so variable, because I was seeing it in my own studies and of course in all of the research that I was reading. And, you know, I think it really is important to understand this from this perspective, which is that, of course, we have our genotype, and that really is important in determining some responses. But in addition to that, our genotype is affected by early imprinting, both intrauterine as well as during early childhood influences. And then over time, we have, you know, all of this interacting with the various environmental factors that we are exposed to throughout our lives. So lifestyle, of course, chronic diet, but also acute diet. Sometimes we have irreversible tissue damage that can permanently change certain things. Exposure to toxins, whether chronic or acute. There's a mental, emotional health component. Of course, fitness level is important, our overall health state. There's, you know, things that cycle on a monthly cycle. For example, in women, the menstrual cycle. There's things that cycle on a daily cycle. Cortisol levels go up in the morning, they go down throughout the day. And then, you know, there's these kind of hourly fluctuations that happen as well. So there's so much variability inherent in not just between individuals, but even within an individual at different points in time. So it really is a very, very complex picture that we're thinking about when we look at what is someone's phenotype with regard to nutrition and how they respond to different interventions. So as I was mentioning, I found out really early on that there's these huge differences in response. So to different nutritional interventions. So for example, you know, here I was early on interested in the effects of genotypes. So for example, the ApoE3 versus ApoE4 genotype. And response to different types or levels of fat in the diet. And in this case, what they were studying was a low-fat diet versus a high-fat diet versus a high-fat diet with DHA. And they found that in the E4 carriers, there was a much larger decrease in triglycerides. But this mean response was, you know, 25%, whereas the actual range of responses, even within that genotype, was everything from a decrease of 75% to an increase in 117%. So it really is super shocking sometimes when you see just how much variability there is between people. And then here on the right, you can see, these were just a group of young, healthy, white male subjects. So we're talking about really kind of narrowing things down on all the parameters and factors that we think are relevant in determining variability in response. And still, even when you have this quote unquote homogeneous group of people, you still get these really high amounts of variation. So this is just simply looking at total triglycerol concentrations after a high fat challenge. And you can see at 4 hours how much variability there can be in the total triglyceride content circulating in the bloodstream. So these are just two really simple examples showing how people are very different. So this is why we really need precision nutrition, because we have to somehow start to go beyond looking at just averages and start to look at individuals. So this concept of precision nutrition, actually, believe it or not, the first paper was published in 1961. And it's been, kind of brewing under the radar there for quite a long time. And then it was right around the 2010s that it really started picking up. And now there's a lot more interest in this area. So the ZV study shown here was one of the first, and I'm embarrassed to say that, they are not from a nutrition department. They are actually, I think they were both either microbiologists or geneticists who did this study, but it was really well done. And what they figured out was that we were finally at a point where we had tools in our hands that we could use to really personalize nutrition. And the first one that they did was using continuous glucose monitoring to basically try to lower glycemic responses to foods. And they did such a great job with this. It was published in Cell, and they're now doing even bigger and better studies in this area. And they actually launched a company to look at people's glucose levels using CGM, or continuous glucose monitoring, to try to personalize diet. And finally, the NIH has also recognized the importance of this area and recently has awarded $170 million for multiple centers around the country to study precision nutrition. On the practice side, or in industry, as often happens, industry is a little bit ahead of research, and there's been lots of companies, I think by my reading more than 400, that are involved in some way in precision nutrition. So there's genomics-based companies, microbiome-based companies, and various companies that look at either metabolomics or other blood work-based outcomes or measurements to recommend a personalized diet to different individuals. And, there's lots of people who feel like we're there and we're ready for this. And then there's lots of people who feel like, yeah, we really should be careful because we don't quite know yet whether what we're doing is making any sense. So that's a bit of a controversial area, whether all of the companies that have spun out with these ideas are really doing people good, or if it's a little premature. So the main topic that I'd like to discuss today is, how do we go about actually being more precise in nutrition? And what I'd like to convey to you guys is that I think that it really has everything to do with discovering the proper targets. And then once we have these proper targets, we can form, the measurements around them, the interventions, and also use them to actually then personalize nutrition for individuals. So I'll do this through describing 3 specific examples, one having to do with the gut microbiome, another having to do with the omega-3 fatty acids, and then the last one is in terms of HDL. And that one I say for last because I think it's sort of the most complicated of the 3 stories. So first of all, with the microbiome, I'm sure you're all aware that the gut microbiome is involved in many aspects of health, that dysbiosis is associated with a number of different diseases, and we pretty much know what an unhealthy microbiome looks like. So generally speaking, regardless of the disease or condition that we're looking at, the microbiome is usually consistent with having increased pathogenic microbes and decreased beneficial microbes. But the problem really has been in the fact that we don't have a healthy microbiome target, because there are many different healthy microbiomes. So there were some early studies early on from, based on the Human Microbiome Project that basically found that there's lots of different ways to have a healthy microbiome, differences in species and all of this really weren't the way to tell apart healthy microbiomes from each other. So how do we actually improve the individual microbiome? Well, I'll tell you one story here about a bad guy. In this case, it's Bilophila wadsworthia. There was a really beautiful paper published in Nature by Suzanne Devkota back in 2012. They found that when they fed susceptible mice, these were IL-10 knockout mice, with a diet that was really high in milk fat, so high in saturated fat, that these mice had a bloom in the Delta proteobacterium Bilophila wadsworthia. So this guy's a really bad guy. It basically induces colitis. It's super pro-inflammatory, and it can induce inflammation even without changing the overall structure of the gut microbiome. So it can be present at really low concentrations and do a lot of damage. And, the question really is, are we all at equal risk of this type of thing? Or, does this mean that we should all eat low-fat diets because otherwise we're all going to have lots of Bilophila wadsworthia and get colitis? That's what we wanted to know. We were interested in, can short-term diet intervention reveal who is susceptible to a Bilophila wadsworthia bloom? So we actually conducted a short and small study. But I feel that it's really the key here is, the study design. So this crossover study design is a very powerful way to discover what's going on in individuals. And you don't need 600 people because each person is just being compared to themselves. So each person is their own control. And essentially, it was only 4 days long. The reason that we did this study with such a short timeline was because I was actually really interested in this question. Could you use something like this in the clinical setting? Could you put someone on a diet for 4 days and then have them come back and then put them on another diet for 4 days and then have them come back again and discover something that you could then tell them specifically, hey, look, here's how you responded to this diet. Here's how you responded to this diet. Here's what I think, in terms of whether this is good for you. It's really easy to adhere to something for 4 days. So it has a lot of advantages. But we wanted to know, is this really long enough to be able to see anything? So we basically put people on kind of an extreme different diet. Here we put them on either a fast food diet or a Mediterranean diet. And you can see the overall composition of the diets was very, very different. On the fast food diet, they were eating 160 grams of fat versus 95. The saturated fat percentage was 20 versus 4. There was no omega-3 in this versus 2 grams per day in the Mediterranean diet. Much higher cholesterol and much, much lower fiber, only 9 grams per day on the fast food diet and as high as 66 on the Mediterranean diet. And we were very, very careful. The females were all tested during the same phase of the menstrual cycle. We took all of these different anthropometric. We asked the night before, did you have anything to drink? Did you take a Tylenol the last 24 hours? Have you? Very, very detailed inclusion and exclusion criteria so that we could really make sure that we were looking at people's responses to diet in a very stable baseline state. And this is super critical. Otherwise, you don't really know what you're measuring. So this is just to show you what was on the meal plan. The food was provided to the participants, so we knew that they ate all of this. They also brought back their containers, and we knew exactly how much and what they ate. Actually, in the case of the fast food arm, they were given gift cards with the specific dollar amounts to buy a specific meal at that particular location with the tax included and everything so that we knew exactly what they were purchasing and what they were eating. So very simple frosted Pop-Tart with, like a cheeseburger with fries twice a day. So once for lunch and once for dinner. No one really had any trouble eating this diet, although we did try as a pilot study to do this for 7 days, and people were like, nah, I'm not doing this. By day 5, they didn't want to keep going. So we made it a 4-day study. And then on the Mediterranean diet arm, you can see it was a very typical sort of high nuts and vegetables, leafy greens, lots of extra virgin olive oil, very low in, there was definitely some tuna and, sources of protein, but no red meat, that kind of thing. So very kind of nice, healthy diet. People actually had more trouble being on this diet and were full and had more trouble finishing their full day of meals. These were isocaloric, same total calories for each day. So when we looked at the microbiome, what was really striking, and others have observed something very similar as well, is that some people are just really resistant to change in their microbiome in response to diet. So for example, this individual here, all 4 time points, they looked pretty much exactly the same. Same with this individual. And then there were other people whose microbiome was much more flexible and changed a lot more in response to the microbiome. And then, we saw some things that were expected and that are universal changes that have been described previously by others who have fed similar high fiber versus, low fiber diets. And essentially we saw what we would expect to see, which is that fiber fermenting microbes were increased after the Mediterranean diet, and protein and fat degrading microbes were decreased after the Mediterranean diet and typically increased after the fast food diet, which is exactly what we would have expected. But here's where the interesting piece of the story comes in terms of variability in response. So as you can see here, there were some individuals who simply did not harbor any Bilophila wadsworthia at all at baseline, and therefore it did not change in any way. It didn't go up, it didn't go out, it simply wasn't there. So these are individuals who are not carriers of Bilophila wadsworthia and were unaffected by the diet. And then you can also see that there's individuals who had low concentrations or abundances of Bilophila wadsworthia at baseline in the light blue here, who were very responsive. And when they were on the fast food diet, their Bilophila wadsworthia really increased quite drastically. And then we also had people who actually had fairly higher levels of Bilophila wadsworthia at baseline, and surprisingly had a decrease in Bilophila wadsworthia after the fast food diet, which was not expected because, they were eating 160 grams of fat, and much of that was saturated fat. How's it possible that this microbe was actually decreasing? Well, we took a closer look at the diet records and found out that some people's diets were actually higher in saturated fat before they were on the fast food study. So their normal baseline diet was so high in saturated fat that eating 2, burgers and fries actually, decreased their total fat intake and total saturated fat intake. And we found that Bilophila abundance was directly related to baseline saturated fat intake. So what this tells us is that we can actually, one, detect who harbors Bilophila wadsworthia. We can determine whether someone is hyper-responsive to saturated fat content in terms of change in the abundance of this deleterious microbe. And we can potentially fine-tune the sort of amount of saturated fat and fiber composition of the diet to minimize the abundance of this pathogenic microbe. So I think, this is a good example of, an actual microbiome precision nutrition target that we can go after right away, because we know that this particular microbe has nothing but bad things to do in the gut, and we don't want it there. Some people are hyper responsive to it, others are not. And so now we know how to figure out who that is and what to do. So the next example I'd like to share with you is essentially focusing on omega-3 fatty acids and inflammation. And so I'm sure many of you are familiar with all of this, since there's lots of clinicians in the audience and others who are perhaps involved in cardiovascular disease. There's been so many studies on this topic, and it's really been very well researched, and there's been a lot of large studies lately. But the fundamental story is that it was first recognized, I think it was sometime in the 80s or perhaps even a little earlier, that the Inuit basically have no cardiovascular disease, no allergies, no form of inflammatory disease whatsoever. And basically it was figured out that this was because they are on a very high omega-3 diet. Essentially their traditional diet was, seals and fish. And so that's why they had such high intakes of omega-3. So many studies show that omega-3 are beneficial that they are associated with reduction in cardiovascular mortality and other inflammatory conditions. But it's been one of those fields that's just been plagued with inconsistency to the point where, it's like you're pulling your hair out going, which is it? Does it work or does it not work? There's some studies that actually came out showing that omega-3s are harmful. A recent study, the VITAL study, which looked at 25,000 people, they found no effect on cardiovascular events. And so it's like, what is going on with these omega-3 fatty acids? We know, looking at them mechanistically, that they should be anti-inflammatory, right? So the basic fundamental mechanism is that, they're metabolized into oxylipins through cyclooxygenase, lipoxygenase, and cytochrome P450 enzymes, and then they have all these autocrine and paracrinic effects. They compete with the omega-6 fatty acids for metabolism, so they essentially displace arachidonic acid from being converted to the more pro-inflammatory oxylipins. And then they also have some physical structural effects such that they essentially disperse lipid rafts, which often are associated with a lot of inflammatory signaling. So for example, T cell receptors, B cell receptors, TLR receptors are all lipid raft receptors. And when you have a higher content of omega-3 in the plasma membrane, this type of signaling is reduced. So one of the things we wanted to do was simplistically just determine the response to an omega-3 supplement. We just took 12 healthy subjects and we fed them quite a high dose of EPA and DHA. And we really just wanted to know, how did this EPA and DHA get sort of distributed across the different lipid classes? And what were the concentrations of the different fatty acids across all the lipid classes, but also the concentrations of all of the oxylipin metabolites? So the omega-3 response, some aspects of it were just completely obvious and as expected. So you feed tons of EPA and DHA, and you see EPA and DHA appearing at increased concentrations in various lipid classes. Makes perfect sense, right? Increase in DHA and PC, increase in EPA, just like we expected. We also expected the intermediates of omega-6 metabolism to decrease across lipid classes. And again, we did see exactly what we expected to see, which was decreases in all of these omega-6 metabolites or intermediates across the lipid classes. Great. Then we started looking closely at some of the specific lipid classes, and we were absolutely stunned by the degree of variability and the magnitude of response to the same exact dose of omega-3 supplement. So we had some individuals here whose PE-EPA increased, from like around 100%, and others in whom it increased by 2,000%. With DHA, it was even more surprising, where we actually had everything from a decrease in DHA in the PE lipid class all the way up to, over 150% increase. And then we saw the same with arachidonic acid. Normally, what should happen is that arachidonic acid levels are decreased because, the omega-3s are competing for the same enzymes for metabolism. And so we should be synthesizing less arachidonic acid. So in some people, you saw the expected decrease, anything from about 40% decrease, but in some individuals, there was actually an increase in arachidonic acid. Very strange. And then we saw this really strong and very interesting relationship in the amount of DHA that was incorporated into the PE lipid class relative to the baseline arachidonic concentration in the PE. And we found that some people who have really high constitutive levels of arachidonic acid in their PE are basically resistant to incorporating DHA. Their enzymes just don't really change what they're putting into the PE. They're just keep putting arachidonic acid in there regardless of how much DHA there is around. So there's some really interesting things going on here in terms of lipid metabolism that I feel like we still don't fully understand. And that could be explaining some of the really high variability in response and why some people seem to have such a strong effect, anti-inflammatory effect from omega-3s and others do not. So another angle from which we looked at this was whether some individuals, were better metabolizers of the plant-based precursor to omega-3. So as you may know, you can either get your EPA and DHA straight from fish oil, preformed, or you can, in fact, take in alpha-linolenic acid and then potentially convert it to EPA and DHA. There's some studies that have found that this is happening at a very low rate, this conversion. But we really wanted to see how much was the variability in this conversion to that. So we conducted a study where we just had 3 individuals consume the same exact shake 3 times to see how consistent this response was over time. And we deliberately did not control for diet and lifestyle because we wanted to see, is there something about this that's really consistent in people, and it has nothing to do with what they've been eating or doing or what season it is or anything like that. So what we found was, and perhaps we were just really lucky here because we had one person who simply did not seem to convert the alpha-linolenic acid to any of its downstream metabolites really at all. So we monitored 18-3 or alpha-linolic acid and all of its downstream metabolites, as you can see here. And this particular individual just basically did not synthesize any of the downstream metabolites. And we sort of termed this person a non-metabolizer. And then you have these 2 other individuals who are converting it just as expected and are, therefore, we're calling them metabolizers. And you can see this going on down the line through that entire conversion cascade. That, individual C here is simply not making any of these downstream products, whereas individuals A and B are. So what do we learn from this? We can measure omega-3 concentrations. Many do so in red blood cells to assess omega-3 status. But I think we can do even more than that and understand not just status, but also, how the omega-3s are distributed across the lipoproteins within the different lipid classes. We can determine appropriate dose of omega-3 as EPA or EPA plus DHA to modify the content of specific EPA and DHA pools. So it may be the case for certain conditions, a particular pool, it might be the phosphatidylethanolamine and how much DHA it has in it that's really critical for a particular disease condition. And so we can, in fact, look at these things. We can also determine whether an individual can obtain a high enough omega-3 by eating a plant-based diet or plant-based sources of those, or if they need marine omega-3 sources. So those individuals who can, in fact, convert alpha-linolenic acid to EPA and DHA could get their omega-3s from flax and walnuts and et cetera. Whereas those individuals who simply are not metabolizers, they're just going to have to take the EPA and DHA directly. So I think there's still a lot more we can do in the omega-3 space to really try to clarify who might benefit from what kind of supplement at what kind of doses and for what kind of conditions. And then my final example here is what I consider to be sort of even the most complicated version of this story. I think there's a lot of other low-hanging fruit that we're all going after in the precision nutrition centers and through other large studies to dial in things like, what kind of foods raise glucose in which individuals through the continuous glucose monitoring and other things like this that are really going to lead to some answers very quickly. But then, I wanted to show you sort of at the other end of the spectrum that there's some other fascinating stories going on in terms of precision nutrition targets. So we got involved in Alzheimer's disease because it's sort of one of these diseases that we really have to figure out how to solve. It's affecting a growing proportion of the population, and the drugs that are targeting late stage processes once dementia and brain volume loss have set in are really, some have finally sort of shown a little bit of improvement, but it really feels like it's a little bit too late because we're starting the interventions way over here when we know that the disease is starting to develop, decades before that. So we really need long-term treatments to prevent cognitive function loss and dementia onset. And if that's the case, if we're talking about interventions that need to be started, in individuals who are in their 40s so that we can prevent something that could be happening in the 60s and 70s, it really has to be something that is safe. So nutritional approaches are actually a great way to do this because, of course, we all eat. And, there's nothing safer than a nutritional approach compared to, drug targets if we need to do this for decades. So I want to start by sort of convincing you, I suppose, that cholesterol is a clear and critical intervention target for Alzheimer's disease. So first, we do know from some really beautiful mechanistic work that's been done over the last 10 years or so, that cholesterol accumulation in the neuronal membrane is in fact directly linked to increased amyloid beta production. So beta secretase and gamma secretase co-localize with amyloid precursor protein on lipid rafts. And when there's more cholesterol in the membrane, you have more of this lipid raft co-localization and therefore a higher production of this amyloid beta 42 toxic peptide. We also know that cholesterol accumulation in immune cell membranes leads to increased pro-inflammatory activity and activation. As I mentioned to you before, these lipid rafts are co-localizing proteins, for example, TLR4 here, that then forms a dimer, which is then activating the downstream signaling from that. Whereas when there's less cholesterol in the membrane, you have reduced inflammatory signaling. And we do know that activated microglia are associated with pathology in Alzheimer's disease. And then finally, ApoE is the strongest Alzheimer's disease genetic risk predictor. And ApoE, the protein, is in fact the main transport for cholesterol in the central nervous system. So it actually transports cholesterol both to cells, for example, delivering it from astrocytes to neurons, but it also actually is the same protein that's used for effluxing or clearing excess cholesterol from cells. And it's also been shown that ApoE is involved in amyloid beta clearance from microglia. So there is some pretty compelling evidence that plasma HDL are involved in Alzheimer's disease. So HDL particles, as you probably appreciate, are involved in cholesterol efflux. It's not clear whether they actually cross the blood-brain barrier, but even if we sort of put a black box there and we say, we're not sure whether plasma HDL particles actually cross the blood-brain barrier or not, let's kind of set that question aside for now because there's some controversy around that. But even if they don't, there is a link between plasma HDL and central nervous system function. So high plasma HDL cholesterol has been associated with reduced risk of Alzheimer's in cognitively normal elderly individuals, even after adjusting for known risk factors. It's also been shown that during midlife, HDL-C is negatively correlated with the onset of dementia 20 years later. And it's also predictive of longevity. Conversely, low plasma HDL cholesterol is found in many of the Alzheimer's comorbidities. And also Alzheimer's patients have lower serum ApoA1 levels. And then finally, and perhaps most convincingly, there's been several animal studies that have shown beneficial effects of increasing plasma HDL. So for example, overexpression of peripheral ApoA1 in a mouse model led to reduced amyloid burden, reduced neuroinflammation, and preserved cognitive function. And likewise, injecting reconstituted HDL particles peripherally actually reduced soluble amyloid beta in the brain. So we might be thinking to ourselves, let's increase HDL. Seems like a pretty obvious solution here, given what we've observed. Well, the trouble is that we don't know exactly what to do or how to do it. So yes, more HDL is better, but not too much. So there are some studies that have come out showing a U-shaped relationship between HDL cholesterol and mortality risk. So there is this kind of sweet spot of a good amount of HDL, but then at some point, too much HDL might be a bad thing. Recently, there's been some discussion about how perhaps alcohol intake is actually skewing that relationship, but we're not really sure yet what's going on. Also, we do know that the amount of HDL doesn't necessarily correlate very well with function. So measuring HDL cholesterol really only explains about 40% of the cholesterol efflux capacity of the particles. So really, just measuring the amount of HDL, especially just as HDL cholesterol concentration, really isn't telling us enough. And then finally, more and more studies are emerging showing that HDL is really complex, that there might be as many as 16 different subclasses with distinct size, protein, lipid, RNA cargo, and lots of different functions. So it's not just this simple cholesterol efflux function, but there also might be anti-inflammatory, anti-proteolytic, antioxidant, and lots of other functions that HDL particles confer as well. So there is growing evidence that cholesterol efflux capacity is an important outcome and that it's associated with cardiovascular disease outcomes. So this has been repeated now across several large cohorts, the Dallas Heart Study, the MESA study, the Jupiter study. Just one moment. Apologies, I just wanted to close the door and reduce the noise from the other side of the room here. And we wanted to know, does HDL cholesterol efflux capacity play a similarly important role in Alzheimer's disease? So there are some studies that were done previously that showed that HDL cholesterol efflux capacity is indeed reduced in Alzheimer's patients compared to controls. So this was a study that had 39 patients and 20 controls, but there was some really important shortcomings in this study. So the Alzheimer's patients were significantly older, and whereas, maybe there's not much of a difference between a 20-year-old and a 30-year-old, but there is a really big difference between someone who's 74 and someone who's 84. And then ApoE genotype was not measured here, and this is a really critical piece because ApoE4 increases risk for Alzheimer's disease, and the population prevalence of ApoE4 is about 9 to 23%, but the prevalence of ApoE4 in patients, in Alzheimer's patients, is much higher. It's greater than 50%. So it's very possible that there was quite a heavy skew toward ApoE4 carriers in the AD group here. So our group did a really small pilot study as well. We convinced ourselves that there might be something here, and we went after a much larger study to look at this. So we obtained samples from the UC Davis Alzheimer's Disease Research Center, and we were able to look at a total of 194 individuals, controls, individuals with mild cognitive impairment, and individuals with Alzheimer's disease. And we had ApoE3E3 and ApoE3E4 patients in each diagnosis group. And we had approximately equal distribution of males and females, and everyone was pretty much age matched across all of the groups. So we isolated the HDL particles. We have a technique where we isolate the HDL first by ultracentrifugation, followed by size exclusion chromatography. And we then took those isolated HDL particles and we measured cholesterol efflux capacity, as well as lecithin cholesterol acyltransferase, or LCAT activity. So LCAT activity is basically measuring the ability of the HDL particles to mature into the sort of larger spherical HDL particles that can carry more cholesterol. And the cholesterol efflux capacity assay is basically just measuring how much the HDL particles, how much of the fluorescent cholesterol that was incubated with the cells can the HDL particles pull out. And I'll just note really quick here that the HDL were dosed as amount of protein. And so what we found was that there was no difference in HDL cholesterol efflux capacity by diagnosis group when we did not account for ApoE genotype. So there was a lower LCAT activity in the MCI cohort, didn't quite reach statistical significance in the AD patients, but there was no signal at all in the cholesterol efflux capacity. But as I mentioned, we know that ApoE is important in lipoprotein metabolism and could be heavily influencing these results. So as I mentioned, ApoE4 carriers are at much higher risk of Alzheimer's, and there's also really important biology going on here So ApoE4 has much higher or stronger binding to LRP1, and it has stronger binding to VLDL versus HDL particles compared to ApoE3. So lipoprotein profile is really heavily influenced by ApoE genotype. When we looked at just genotype, ignoring the diagnosis groups, we found that indeed the ApoE3/E3 carriers had higher efflux capacity and higher LCAT activity than the ApoE3/E4 carriers. So then when we further stratified by ApoE genotype and diagnosis group, we found that ApoE3/E3 carriers, the patients had lower cholesterol efflux capacity. Again, didn't quite reach statistical significance in the AD group, but definitely lower in the MCI patients. And the controls, again, also had higher LCAT activity. But we found a surprise here. And that was that the ApoE3/E4 patients had higher cholesterol efflux capacity. There was no difference in the LCAT activity, but they had higher efflux capacity. So we were confused by this, and we really wanted to know, why was the HDL-CEC higher in the ApoE3/E4 patients versus controls? So the first thing that occurred to us is that it was possible that the E3/E4 patients had more small HDL particles. So if you go through this thought exercise for a second, you realize that, and I'm just showing an extreme case here of someone who has, only 10% small HDL and 90% large HDL versus someone who's got mostly small and very few large HDL. What you notice right away is that, if someone has not very many small particles, you're only putting on a total of, say, 20 HDL here. Whereas for the same protein amount, if someone has tons of small HDL particles, you're dosing that well with more than double the amount of HDL particles. So we wanted to look closely at the size distribution of the HDL particles, and my student, Jack, developed this method to actually size particles using transmission electron microscopy. And we basically had to develop a protocol that would allow us to look at, large numbers of clinical samples, but to do so in a way that was going to be automated so that he could image, thousands of particles for each sample so that we could get a reasonable estimate of the distribution of the size. And what we found was that indeed in the ApoE3/E4 carriers, the Alzheimer's patients had significantly smaller HDL size. And this is just looking at average size. Then we decided to look at, more deeply, because now we had tons of information about these HDL and their size distribution. We wanted to look at, what was the real change? Okay, so the average size was smaller, but what was the actual change? And we figured out that actually the ApoE3/E3 carriers didn't have much going on in terms of differences in particle size, some things that came up as statistically significant, but nothing very striking. Whereas the ApoE3/E4 carriers, the patients had higher levels of these smallest HDL particles and lower concentrations of the medium and larger HDL particles. And in fact, when we looked closely, what we realized was that it wasn't so much that the patients had higher levels of the small, it was that the controls really had lower levels of the small particles and higher levels of the medium and large HDL particles. So what does this mean? Well, first, we wanted to see if there were any correlations with things that made sense to us. So what we found was that the small HDL were positively correlated with BMI, and the medium and large were negatively correlated with BMI. And perhaps more importantly, the small HDL were negatively correlated with a measure of cognitive function. Here it was verbal memory score. So many of you might be familiar with this pathway, which is basically in the context of high triglycerides. The enzyme CETP is basically transferring triglyceride to HDL particles and cholesterol back to the ApoB proteins. And the result of this is you basically form these small dense HDL. And so we are thinking that in fact, in order to not have cognitive impairment by average age of about 72, if you're an ApoE4 carrier, you need to be in a healthier metabolic state. And this disruption in HDL particle size distribution could either be just a simple readout of the metabolic state, right? It could be just that these individuals have the typical sort of metabolic syndrome hallmarks, and the small HDL is just one of those features. Or it could be that this is somehow directly related to pathophysiology. So it could be that this loss of large HDL is somehow actually involved, and we're pursuing this as an interesting potential target. So we figured out that the ApoE3/E4 patients likely have disrupted HDL particle size distributions, so they have this more small HDL and less medium and large HDL that's related to obesity or metabolic syndrome. But what about the ApoE3/E3 patients? We were really interested, could it be that they're damaged? Is this why they have less functional capacity? And so we actually conducted some basic in vitro experiments. We just made one giant batch of HDL to determine which chemical modifications of HDL really have kind of the strongest impact on cholesterol efflux capacity. And could things like lutein or zeaxanthin, which are transported by HDL particles, actually improve function? These are antioxidant carotenoids. And lo and behold, it looks like exposure to low pH, various oxidizing agents, so we tried 3 different ones, and fructose all decreased HDL cholesterol efflux capacity really strongly by as much as 75%. And then, as you can see, lutein and zeaxanthin here actually did improve the cholesterol efflux capacity of HDL particles. And then we also found that low pH oxidizing agents and fructose also decreased LCAT activity, not as strongly, but still there was a decrease in LCAT activity as well. So HDL particles as a precision medicine target for Alzheimer's, I think is possibly a promising direction. We think that probably the E4 carriers kind of bear more of a classic signature of increase in small HDL, loss of medium and large HDL, characteristic of metabolic syndrome, and that perhaps management of metabolic health, hypertriglyceridemia, insulin resistance might be even more important in ApoE4 carriers. Whereas in the ApoE3/E3 carriers, they seem to have decreased HDL functionality without major changes in particle size, which kind of leads us to believe that perhaps this is due to increased damage and that management of oxidative stress and perhaps supplementation with antioxidant, but specifically, these carotenoids that are specifically transported by HDL particles might really be important in this group. So I've shown you some examples of how precision nutrition can already be implemented in practice now. Some examples of ones that are still kind of up and coming. We're not really quite there yet. That precision nutrition approaches have the potential to improve individual health and decrease specific aspects of disease risk. I hope I've also convinced you that study design is actually really important for discovering precision nutrition targets. You really have to make sure, especially if you're using metabolomics or other omic tools, that you're very tightly controlling, the environment under which people are coming in for that blood draw, and or that you have multiple blood draws so you can compare people to themselves. The pressing question is how to integrate this into clinical practice. And I think that's probably going to be the biggest question mark going forward. So thank you all for your attention. I'd like to acknowledge, of course, the financial support and all of my lab members and collaborators, and I'm happy to answer any questions.
Angela, that was fantastic. I will clap on behalf of all of the people who are online.
Thank you.
That was great. There's a bunch of questions in the chat. Perhaps I can just start with the first high-level one.
Yeah.
In the first 2 examples, and I asked because there's probably some computational biologists there always is, who are doing artificial intelligence and stuff. In the first 2 examples, you talked about challenges in patients and then looking pre and post. So one was a dietary intervention, one is a supplement, and then, who's responding, who's not responding. And you showed it in very small numbers of patients, each of them being their own control. But if you did that at scale, you could then do predictive modeling for who's going to have a good response or have a poor response. Do you know the groups? Are there groups doing that at scale? These small term interventions, like you said, like small term dietary interventions, like in the All of Us program or something?
Yes. So from what I understand in the precision nutrition centers, which I'm actually not involved in one, what they're doing is both sort of larger cohort studies where they have people coming in and they're going to have single time point measurements, metabolomics, proteomics, everything you can imagine. And then for a smaller group of people, it's still much larger than our studies, but hundreds of people, I think, rather than thousands or tens of thousands, they're going to have 2-week dietary interventions where, and I don't know what they've decided on in terms of exactly which dietary interventions they're going to be, because they spent, the first year debating and deciding which diets they were going to intervene with. And then they were going to see some responses pre and post. And there have been studies that have done this. So the PREDICT trial was a study that did that, where they took a pretty large number of individuals and looked at them pre versus post dietary intervention to look at, how the metabolome changed. My feeling on this is that is going to be informative, and that we're going to discover a lot of things by using some wonderful computational techniques on those really nice big data sets. But that, we can still also learn things from some smaller, very, very well-designed studies, even though they're smaller. Because in the smaller studies, you can really control everything. There's an upside and an outside to everything. You can do things at scale, but maybe they're not quite as tightly controlled. For things that are really tightly controlled, there's just so many parameters and there's so much one-on-one time with every single individual that you really can't do it at scale. But we can still learn things from those small studies.
Totally. And so a question and a comment from the chat. The question from Akira Sekikawa asked about the conversion rate of ALA to DHA, and noted that isotope studies generally report that the conversion rate is pretty low, 3 to 5%, except for pregnant women. Do you know the conversion rate in your study?
We did not do a stable isotope study. So again, it's kind of another perfect example of what I just described, which is, stable isotope studies. Those are done in, 6. An N of 6 is an amazingly high number for a study like that. So although I'm not disputing those findings at all, what I will say is when you look at 6 people, you don't necessarily know how representative that's going to be of a larger population. And so I wouldn't be surprised if we did a stable isotope study in 1,000 people, that we would find some people who converted the heck out of ALA and some who really didn't. I think it's still safe to say that the conversion is not very high on average, but I think that some individuals are definitely making a lot more EPA and DHA than others. Yeah,
makes total sense. So Stephen O'Keefe said, "Great talk. You're right. Nutrition is complex." That's the comment. And then he said, "The Inuit now have one of the highest risks of NCDs in the world, but their N3 intakes and levels remain very high." Can you comment on that?
I don't know, the details of what's going on with those populations. I did have a colleague who was working with an Inuit population. This was probably about 10 years ago. But what she was finding was that their intakes of omega-3s were not high anymore. That their diet had completely changed from that traditional diet that was high in fish and seal and all of that, to essentially a really junky kind of Western diet where they were, essentially eating just as much, corn and processed food as any average American, anywhere in the United States. So I think that the main, issue probably is a really large shift in their overall diet. And even if their omega-3 intake is higher compared to other Americans, because they maybe still have some traditional meals or holidays where they still eat traditional foods, and that might boost their omega-3 intake to higher than your average American in other parts of the US, still their overall diet appears to be very high in omega-6, very sort of unhealthy compared to how they were eating before. And they might be an inordinately higher risk for, if those populations were eating really high omega-3 diets for tens of thousands of years, then really having a large drop in that and a large increase in omega-6, which they were never seeing before, would maybe have a stronger effect, and it might be a bigger problem for them than for other individuals with a different genetic background.
Yeah, super hard to control for this. I think that's what Stephen was saying at the start, that it's a complex subject. So the next one's from Katie Polek, a question about precision. Thanks for the great talk. Cholesterol efflux capacity is often assessed using macrophages, as you've done here. Do you think this accurately represents CE capacity by other cell types, given variability in ApoE receptor expression? How might this variability by tissue cell type affect disease progression? A very interesting precision question.
I love that question. Yeah, the reason we all measure it in macrophages is because no one besides cardiologists was interested in HDL, right, at first. Yeah. And so there's a lot of even how HDL particles are thought of as good cholesterol, and why we measure them in macrophages is because the first discovery of their function was in regard to atherosclerosis and removal of cholesterol from atherosclerotic plaque. I agree that it's really important and interesting to look at how HDL particles interact with other cells in the body, because they do efflux cholesterol from pretty much all of the peripheral tissues, and that's the only way that a peripheral cell of any type can get rid of its cholesterol. It can't break it down in any way. It has to efflux it. So I totally agree that looking at the function of HDL and efflux from other tissues is really important. Yeah.
And so a question from Jay Bahari. I was going to ask you this at the end, but it's good that it comes up now, because I think it relates to your last point you made about clinical implementation. So he said, great talk. In clinical practice, the most prevalent chronic metabolic disorders, DM, obesity, fatty liver, etc., are due to excessive intake of refined sugars and highly processed food. Is there any evidence that precision medicine can help in this large group of patients over and above focusing on decreasing intake of these foods? So other ways to target this clinical intervention?
What a great question. And I've sort of deliberately left that to others to solve and focused on my own little niche area, because that's my strategy for obtaining funding. But yes, I think that it could be really helpful, because in the end, it doesn't matter how you reduce the excess nutrition, right? The problems of insulin resistance, type 2 diabetes, metabolic syndrome are problems of overnutrition, right? And you could argue that it's a combination of overnutrition and underexercise, or not enough physical activity, but really the fundamental issue is overnutrition. So however you can reduce that overnutrition, you will have an improvement, right? However you lose the 10% of body weight. The question is, which one do you choose for which individual, right? Which one will actually work for that person in terms of long-term sustainability? And so, yeah, I think it would be very useful to use some of these principles of precision nutrition to help people discover what works best for them, because you don't necessarily want to exacerbate something, as you're trying to improve something else. So, let's say ketogenic diet, is that going to work for everybody? Probably not. I would argue that there's some people there who would really not benefit from a ketogenic diet, even though it might really lead to convincing weight loss, because it'll raise their LDL cholesterol astronomically and, increase their heart disease risk. But then there's other individuals for whom that's really not a problem. I've personally had clients who, have type 2 diabetes. We put them on a ketogenic diet. Their lipids are not high to begin with. Their lipids don't go up, but their glucose levels dramatically drop. I mean, the variability in terms of metabolic response to these different diets is really amazing. It's almost like interspecies level differences. It's kind of unbelievable. So yeah, I think that it could really help. Especially in terms of sustainability and helping for people to actually stick with diets that work for them.
Yeah. And so a question about the AD study you talked about. What's the mean age of participants in your research about HDL and AD? I'm also wondering if you think we can generalize and apply your current findings among midlife participants.
I would love to be able to answer the latter question. In fact, I'm going to propose that in a grant. Right, because we want to know if this is something we can do with 40 and 50-year-olds so that we can prevent Alzheimer's disease. It would be a really great way to go. The average age in that particular cohort was it was either 72 or 74. So everyone was kind of in their late 60s and 70s.
And your thoughts on measuring LDL-HDL ratio or HDL or LDL alone in the context of Alzheimer's?
I think every biomarker tells you something, right? We just have to be mindful of how much any given biomarker can tell us, right? And so I do think that some of these ratios are very useful, quick tools for giving you some kind of quick readout of something, whether maybe it justifies looking further and doing more complex, more detailed tests. I don't think that just because a marker is easy and cheap and easy to get, that it's necessarily useless. It could be useful to give you kind of like an early indication of whether that's a direction in which you want to do more analysis. But I do think that what we're realizing with HDL is that if you just look at HDL cholesterol concentrations, okay, let me take that back. Even that can tell you something, right? Because low HDL cholesterol is in fact part of metabolic syndrome. It's part of how metabolic syndrome is defined. And so it's probably a great readout of long-term hypertriglyceridemia. But HDL cholesterol alone doesn't seem to really tell us things in other contexts. So in this context of high triglyceride, because of that really well worked out mechanism with CETP, it can tell us a lot about what's going on in that context. But HDL cholesterol alone doesn't tell you much, in non-metabolic syndrome or non-hypertriglyceridemia kind of instances. So there, we really need to do more analyses, looking at, HDL particle size distribution, efflux capacity, antioxidant capacity, other things. We're still really working on that. What else do we measure in HDL and how do we begin to define what's going on with HDL? That's like the most complicated of all of the examples I showed, I would say. We're kind of the furthest away from knowing what to do there.
Well, that's great. I think we will wrap it up there. I think it was a fantastic hour of, the basic science through the translational science all the way through to clinical implementation. I think that was fantastic. I think that people enjoyed it on the line. And Angela, I want to thank you once more for taking the time to do this.
Thank you so much. Really appreciate it.
Bye.
Take care. Bye-bye.